Maxam-Gilbert sequencing method, or chemical cleavage sequencing method, is a first-generation sequencing method. Cleave specific DNA molecule by certain chemicals, and subsequent cleavage at specific bases such as guanine, and adenine in guanine, and cytosine and thymine, which corresponding to radiolabeled fragments, as mentioned in next slide. Maxam-Gilbert sequencing method. Initiate by DNA extraction, and denature double-strand DNA molecule to single fragments, and radiolabel single fragments on 5 ends using gamma-32P, as mentioned in next slide. After that, cleave single fragments at specific positions by using 4 chemicals reactions to generate breaks in specific nucleotides, as follow. Tube 1. Contain dimethyl sulfate to break guanine nucleotide. Tube 2. Contain dimethyl sulfate and formic acid to break purine nucleotides. Tube 3. Contain hydrazine to break pyrimidine nucleotide. And Tube 4. Contain hydrazine and sodium chloride to break cytosine nucleotide, as mentioned in next slide. And incubate 4 reactions tubes with piperidine to break modified strands, and subject reactions side by side in denature polyacrylamide gel electrophoresis to differentiate fragments size according to radiolabels, and autoradiography gel on X-ray film to read gel bands from bottom to top, as mentioned in next slide. So, bands present in cytosine and pyrimidine nucleotides. Refer to cytosine nucleotide. Bands present in pyrimidine nucleotides. Refer to thymine nucleotide. Bands present in guanine and purine nucleotides. Refer to guanine nucleotide. And bands present in purine nucleotides. Refer to adenine nucleotide, as mentioned in next slide. Therefore, Maxam-Gilbert sequencing method. Use purified DNA directly in sequencing process to identify DNA modification without require PCR, and consider accurate and suitable in DNA footprinting studies. On the other hand, Maxam-Gilbert sequencing method. Consider labor and costly and time-consuming method, and require hazardous chemicals as radioactive labels, and read short sequence length, 50 to 500 base pairs. Finally, Maxam-Gilbert sequencing method. Use in DNA structure studies, and DNA modification studies, and DNA interaction studies, and DNA footprinting studies, and DNA fingerprinting studies.